Move the one on the bottom. Suddenly it's a puzzle. Oh, it could be. Maybe I'm dumb. All right, we gotta plan our angle of entry, I think. Well, I just saw the, oh my God, the title of this level, Wiggler Boners. What am I getting myself into? Also, hold on, you guys have seen Bowsette? Where's my Wiglet? Where my Wiglet at? Oh God. Carl's getting demonetized. Oh, just you wait. All right, Wiggler Boners. It looks like the Wiggler has the key in it. Yeah, the Wiggler does have the key in it. All right, guys, I got the keys. I beat the level. Easy. Easy. I didn't beat the level. So that bouncer forces you into Wiggler timeout chamber. Wiggler did not like Snoo Snoo. Wiggler angry now. You need better Snoo Snoo for Wiggler. Alright, what the fuck do I do? I can get this. What does that do? What does this do? Does this do anything? Why is this bumper here? Do I want... I think this is bad because... There's no hole there, right? Yeah, there's no hole right there. I think I know how to cheese this level. But I don't think that's what he wants me to do. I don't know though. I think I might be able to cheese this with Pow Wave tech. What is the other key in? It's the muncher in the top left. So there's a muncher in the top left that also has one. So Pow Wave could probably cheese this, but that's not what I want. I want to try to beat it without cheesing it. Maybe I can... Like, what's the plan here? What's the move? Is it this? What does this do? What if we like squish the Wiggler? Wait, what if we make like a Wiggler path? Is this a Wiggler path type of thing? See what I'm getting at? Oh, I was like thinking maybe it would move the Wiggler. Well, let me try something different. Pow instantly kills enemies, no wave. It is a wave. Pows are waves. It's not a particle, it's a wave. All right, well, we have created a quantum Wiggler. We have created a quantum superposition of Wiggler. What's this gonna do? What the fuck? I'm sorry, I'm trying to help. I know you're trapped between dimensions. I'm trying to help. What if we put this one like... I don't know what the fuck I'm doing. No! Shit! Oh my god. Oh, Pow Wave. Oh, Pow Wave only applies to coins. Oh, okay, interesting. What am I trying to do? I don't even understand what I'm trying to do here. Like, why is there even a shell out there? What's the shell gonna do? How can the shell do anything for me? How am I gonna kill this Wiggler? It's called Wiggler Boners. How does that help? You need to build around the center. Okay, you mean like over here? What are we... Where am I trying to... Yeah, see, like, am I supposed to get it right there? But that doesn't really do much. Let me destroy you, Wiggler buddy. That's not right. Yeah, they're just reset doors. Here, look, like, we can let him come across here, but, like, what's the point of that? How is him going over anywhere better than anywhere else? Like, where do I want the Wiggler to be? Maybe I want him in the corner. That's kind of what I tried, though. I can try to get him in the corner by building this way. I don't know. Might need a small hint. Remember Hello Baby? Oh, you know I remember Hello Baby. Maybe I want the Wiggler to be angry. Hold on, maybe angry Wigglers are better. This is not smart. Maybe angry sex is the sex that we're looking for, is what I'm trying to say. What if we do this? Can he cross that gap? He can't cross that gap, but he should be able to cross this. Look, it's the weird POW text again. The O and the POW is messed up. Why isn't he going across that? Would you? Okay, there he goes. So what if he's across that while I bump him like this? Well, he moves a little bit, but that's it. Alright, that did not... I should have put one more up there or something. I want him to get stuck in the center. What's... Oh, do you mean the center of the... Rob, do you mean... Like, which center are we talking about here? He just went right through the POW. Do you mean the line guide, the track? Center of what? You gotta prep the wiggler? Shit. It could be right here also. I feel like these are our two options. Well, I feel like that's our other option. But what does it do? I'm a little confused. Oh my god, he's so hard. I should not have activated the wiggler. Gotta whisper sweet nothings into the wiggler's ear. Okay, let's not make him mad this time. Got an idea. What if we put it there? Here, I got this. And then we drop it. What was that? Okay, fine, your body has segments and you're so cool, but like, fuck off. What? Oh my god. Come into my trap, please. Maybe it's the head. We gotta work the head. Yeah. That's the problem. All right, wait. I can actually push his ass into that. Hold on. We gotta work the head more. We're not working the head enough. There we go. All right. All right. Oh! We stuck it in the wrong place. We put it in the wrong place. Oh god. Are you guys taking notes? These are very important lessons. You don't just... Alright, you don't just slide it in from behind without asking first. How did that kill me? Hold on, that's not what happened last time. Oh. What is going on? Hold on, let's make it let's hit it. I'm not going here anymore, alright? I'm not gonna say what that symbolizes. Why is this the best thing ever? Alright, we gotta tap that a little bit, and then... What am I even doing? Hold on. I think we gotta, hold on. It's not the right spot yet. It's not the right spot yet. Huh. Maybe I have to... I'm kind of scared to remove the one on the bottom, but I think I might have to move the one on the bottom. Oh yeah, they really like that. You can tell they really like that one. That was a good move. I know exactly how we are going to beat this. And it makes me so happy you don't even know. Oh my god. I don't think I have the right angle. I think my angle's a little off. Oh shit. Okay, this is actually good, I think. No, that's bad. Alright. I just came back, what the fuck is happening? Don't hate what you don't understand. Yes, this is what I want. Alright. Now we've got all the segments. We've got all the segments now. I think the trick is to jump a little bit when you slide it in. You gotta get enough leverage. There we go. Alright, now we finally have been allowed access to the booty. And we have earned our reward. What game am I fucking playing right now? What game am I even fucking playing right now? I know, right? That was like incredible. Is this the Yeah, this is the prequel to Hello Baby, the game of life. Oh my god. World record 6 seconds. It is cheesable. There must be some way to cheese it. Oh god, my fucking cheeks hurt. My cheeks hurt because I'm fucking 5 years old. I don't know what's going on, but there's Wigglers and the level name is You Are Top, She Is Bottom. So, enough said. Enough said. Also, oh my god. All right, we gotta plan our angle of entry, I think. Oh, how was? Is it gonna clip me? Hold on, wait. What's over here? Oh, did I need this? Let me in. I don't know if this has to do with the. I thought that might do it. I feel like I probably need to clip through this. But what's the right way to make it happen? Okay, this has like a 27% clear rate, and I feel like I'm gonna do worse than the average clear rate. That's not good. Hold on, let's get the mushroom. And then let's see what we got here. That almost did something to me automatically. Huh. Jump, maybe? That's not doing it. Duck? Oh, that looked good. What if I jump first? It was really easy. There's nothing wrong with being a bottom. So Potato just slides a level into my DMs, and I'm like, I don't know, it's getting kind of late. And then I read the title, and it's Poo's. Oh wait, shit! I didn't mean to hit refresh again. Throw cheese, and it's Poo Stomp Simulator. hold right and Y. All right, guys, you ready to stomp? Let's go. It crashed the fucking game. For anybody who doesn't know, during SGDQ, during the Mitch Flower Power Grand Pooh Bear Super Mario Bros. 3 race, every time Pooh got behind, no, I'm just kidding, he didn't do that on purpose, he fucking stomped the ground and Mitch's game froze. No! Oh. No!
We gotta hum.
Shake! 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 Shake!
Well.
Shake! Shake! Shake!
So this is the fucking Pooh Stomp Simulator because it freezes the game. My Wii U just crashed. And Potato once again trolls me in ways that I could not have expected or predicted. I think I have to restart. Potato's like, hey, you should do this for the very last level. I have to reboot my Wii U, so give me a second. Oh my god, the screen still shows the game frozen? Oh, I have to hold on, that's like a hard reset. I gotta like hard cut the power, 1 second. Okay. Jesus. Now I just have an error message on my screen. Potato broke my Wii U. Potato broke my... Okay, there we go. Jesus. Yeah, send this level to your favorite streamers and viewer levels. My favorite part was... what the fuck? Ordinarily, please hold the power. Okay. Yeah, I had to force shut it down. Fuck off. Yeah, send this to your favorite streamers. Jesus. Is my controller even working? I don't think my controller's working. It's gonna get banned so fast. Yeah, guys, we gotta submit this to everybody. Imagine getting that level in Super Expert. Oh, God. If your game crashes, you can't report the level. Alright, hopefully my game doesn't die. What? How does it... So one, this must be like a glitched power-up or something? Oh. There's just a Yoshi egg? Is this just that badly programmed? Is there no big glitch here? And it's literally just, if you grab a fire flower right when a Yoshi egg hatches while you have a cape and the game just crashes. There's not even a glitch in the level. Nintendo just sucks. Does it have to do with like you hold a spring with a cape or it's a Yoshi and a Yoshi egg? Hold on. If I just push play in editor, oops, if I just play it in editor, what happens? Good game. Good game, bros. Good game. You have to be in the turning animation with an item while landing on a Yoshi and a fire flower. Okay, to be fair, that's a pretty specific setup. You're in the turning animation with an item while landing on a Yoshi with a flower. Man, I want to know what happens like on a programming level that makes the game shit itself so bad with only that combination of stuff. Remove the spring. If we remove the spring... Oops, that did not remove the spring. Then we won't die. Yeah, it's like that transition of getting on Yoshi while you are turning into a fireball. It's weird. It's really weird. I wonder if it would work if you just happened to do it while a mushroom is cruising by. I think you have the reason that since you have to be holding an item, that makes it weirder. That's probably why, because there's not very many instances where you're spinning while holding an item. In order to spin while holding an item, you have to do something like this, where you're like already spinning and then run through the item. All right, this is Uno Mas pushing to the edge. It's a good question. That's a good idea, actually. Whoa, what happened to that POW? We've got let me move my face over here. Okay, let's see what happened again. I can get this POW. Then there's, what do we need to do? There's only one red coin. We have to somehow obtain this red coin. And that's gonna allow us to go on the key door. So we're gonna get through these bumpers somehow. Can't really do any shenanigans like this. Can I move this launcher somehow? Hitting the POW doesn't actually do anything. I have no idea. Yoshi can tongue red coins, so that's like a thing. What if I'm not supposed to collect this? Like, there's not many options here. I only really have this POW to play with. I'm thinking, like, what if I wedge myself, like, into the launcher? Something weird. Oh! Oh, what the fuck is that? Why does that happen? Okay, someone's got to put this in a troll level because this is too amazing to not use. How am I gonna get out of this, though? Is it gonna kill me? Is it gonna kill me? Wait! That was amazing! Oh my god, someone's gotta put that in a troll level. Going on a magical journey, y'all. Holy shit, that was amazing. Good suggestion, Rob. That was awesome. I'm streaming a little bit longer, yeah. I'm feeling like low blood sugary or something. I don't know though. So I might not stream too much longer. 1112222. Thank you for that gift sub to Myth Marksman. Kenzie B, thank you for the sub. Appreciate it. Professor Plum, thank you for 6 months. That was amazing. Wait, we can beat that world record. We can beat this world record. All right. World record. Why did you bookmark my old levels? Oh, just because when I was looking at your current one, I saw that you had some interesting looking Unomas stuff or puzzly stuff. One of them I wanted to know how you beat it. I didn't look at it, but the thumbnail made me wonder how you beat it. Oh, I'm sorry, Kenzie. Don't worry about it. Anyway, that was awesome. That level was so cool. Potato level. All right. Do we have a potato moss? Do you guys want a potato moss? I'm not sure if I have it. 0%. 0 out of 220. What the fuck are you talking about? What the fuck is 0% clearing at it? Well, excuse me. What are you trying to pull here, Buster? What are you trying to pull here? 0 out of 220? What the fuck is this? What the fuck is this? There's nothing behind Mario. We have a cape. We have a Bizom. I think I know what to do. It's this, right? Pretty sure it's that. Because do we need 2 keys? Hold on, wait, we already have 2 keys. What is this? Set up. Oh, I didn't want to go in that? Yeah, I need to not have a key for the last one. What? Hold on, what is this level called again? 1 key, 2 key. Red key, blue key. I think I only need 1 key. Why did I get 2 keys though? Oh, if I hit it, it gets both. Okay, I get what to do though, I think. I'm pretty sure I just break the block. Hold on, I think it's this. I think it is just this. Oh, would you stop that, please? How do I... Oh, I jump on it and then flick it. Okay, wait. I think it's this. That, and then this. Carl bad. Wait, if this is it, then how have 220 attempts gone by? I missed. Oh shit, that gave me 2 keys too. Oh shit, that ain't it! That ain't it! This? Does that do anything for me? Oh, I wonder if I can, see how I can kind of climb into this shit? Huh. Interesting. Oh, that's all my ideas. Well, that's all my ideas. What do you got, chat? That's... no. What if we do this? That doesn't do shit. That doesn't do shit. Try to hit the bomb so it only breaks the block. It's not collecting the red coin, I don't think. Oh, it could be. Maybe I'm dumb. Maybe what happened wasn't what I thought happened. No, yeah, see, it gets both still. It's not actually hitting the key. It's just when you blow up the block below the key, it collects it. See, it's gonna collect it both, even no matter what, I think. I am pretty out of ideas. There's something behind the wall there, I don't know if it matters. Try to clip the bomb with the vine. I don't know, man. What am I gonna do? What do we, I just don't know what we need to do. Oh, I know what to do. Oh, yeah, no, I know what to do. I don't know how to do it, though. I know what to do, but I don't know how to do it. This is it. No, it's not it. I almost know what to do. If I get the bomb to explode higher up, it could collect the coin, but I don't know how to do that. Yeah, I don't know how to do that. I don't know how to fucking do that. Whoa, what was that? This also looks promising. I don't know how to do it. Thought I had something, have nothing. Thought I had something. Will the block always collect coin if blown up? I think the answer to that is yes. Oh, I can't put anything anywhere. I mean, it's gotta involve the cape, right? But what can the cape do other than this with the bomb? Oh god. It won't blow it up from there, I don't think. Yeah. If I was trying to think of a way to remotely activate the bomb then I could just hold up on the door or something. 1 key, 2 key. Put bomb in yellow, throw. You can't. I tried it. See, I can't, What if I hover with the cape, though? What if I hover right here with the cape? Oh, the thing kills you. Okay. I could spin jump on the bomb right there. Aha! I figured it out. I just don't get what the purpose of the cape is. I have some idea, actually. So you see how if I jump up and then cape spin, it puts Mario higher up. Maybe the bomb is a fucking troll? But I don't know how to make use of that. Actually, I have an idea. Maybe the reach of the cape... Here, wait, I have an idea. I don't know, maybe the bomb's distraction? Watch, let's try this. Maybe it's possible for this, see how big the cape is? Ah! I don't know, that was my idea. Editor, please? Okay. Okay, what's the hint in the editor? Don't really see anything. Maybe it's behind something? What am I supposed to see? Carl blind? I don't see anything. Oh, subworld? What the fuck? What the fuck is that shit? What does that mean? Oh my god, there's 2 red coins. Look, there's 2 red coins. Oh, is the level fucking impossible? Is the level fucking impossible? Was it impossible this whole time? Is that the troll? It's actually an impossible level. It's actually impossible. Damn it, Potato. God damn it, Potato. All right, guys. We're gonna do one more for, it's actually impossible. So there's 2 red coins, but it treats it as 1, and the setup makes it so you can't win with it. Like, if you get even 1 of them, it will kill you, but he was able to upload it because to upload it, there were 2. I have been so deep making my own ROM hack. It takes so much time, it's absolutely ridiculous. Yeah, so I have probably spent like 100 or 200 hours, just all of my free time for like the last month has been working on my ROM hack. So it's ridiculous how long those things take. I will say I really appreciate how easy it is to make levels in Mario Maker in comparison to with Lunar Magic. It's fun though. Dude, I know Barb is like, so if you want to watch like somebody making ROM hacks, check out, again, Barbaros King. I plugged him twice already, but he's literally streamed 700 hours of making a ROM hack. What are you working on? It is a cape ROM hack. It's like really crazy cape tricks. Oh my god, I've been once you get stuff to work, I will say that like once you get stuff to work in Lunar Magic, it is amazing what you can do. Like the possibilities are basically endless. I really hope it gets super diagonal too. All right, we're gonna play a video game and it's gonna work and it's gonna be fun. Are we having fun, everybody? All right, this is Mario's Quest for Mod Chip. I don't think it's a story level. I don't even know what this is. I don't even know what this is. Mario likes. This text is terrible. Watching Carl. Okay. Oh boy. Mario wants to what? Be a mod. Oh, okay. Mario wants to be. Wait, is this like subtly somebody trying to get modded? Oh my god. Want Wait, what should he make? Oh my god, this... Trolls? Troll music? Does that say Uno? Oh, there was text at the bottom. Holy shit. Okay, oh, troll, puzzle, music, story, Uno Mas. Okay, I see. Oh wait, do we have each of these? Wait a minute, is this troll? I want troll. Give me troll. Give me troll. Oh, it is, I think. Wait, no, this is puzzle. This must be puzzle. Hold on. This has to be puzzle. Oh shit, I don't remember how to puzzle. Shit, I have to puzzle. What do... Oh, is this guy still alive? Hold on, is the dude still alive in there? He is, right? Yeah, okay. Okay, suddenly it's a puzzle. Suddenly it's a puzzle. Hey, puzzling good, let's go. It's kind of cool. No! Well, they pretty much nailed it. They pretty much fucking nailed it. I knew it. Oh god. I like how they knew. Alright, how am I gonna hit this? Oh, with the fireball. Okay. Okay, right. So let's let's stand right here. Dude, they know that all of my puzzle levels are actually fucking troll levels. Okay, what if I stand like right here? What if I stand like right here? Okay, Jesus. And then with the vine, we can do that. Yeah. You can like go into it with the fireball. Okay. God. It turns out all 5 of the things are actually just, what the? Carl didn't mod Mario. What? Try again? What the fuck is this? What? I didn't even get the checkpoint. Excuse me. Excuse me. Wait, which one is troll? Is Spikes troll? Was it Spikes, I think? What the hell is this? It's gotta be troll. Oh. 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 Ah, at least we got a checkpoint. LOL CP1! Oh my god. Wait, what does it say? Hold on, does it say something over here? Oh, is that his fucking mouth? Wait a minute, that's the mouth! Oh, I wanna appreciate this. Okay, here we go. That's a fucking mouth! Oh god. Oh. Ban? What does it say? I cannot fucking read this to save my life. Mario. What? Oh, no mod banned. Oh, you didn't see the mouth? Oh my god. Oh, exclamation point ban Mario. Okay, okay. I knew the troll was gonna be the right answer. So there were 3 doors that led back to the other checkpoint. Oh my god, that was hilariously silly. I want to see the other sections though. I like how well they like distilled the essence of my stream down into a few sections. Though this text is like horrifically positioned on the screen. Okay. God, that puzzle was good. Let's try music. Actually, let's try Uno Mas. This is Uno Mas. How did I just get that? What the fuck? What the? All I have to say is this. This is all I have to say. How do I actually do this though? Oh, probably like this. Wait a minute. Oh. Get me out of here! They like they like achieve perfect harmony with each other. Why? I can't stop it. I can't make it stop. Why? I'm pushing Z. Oh, there we go. Okay. But how do I get this one? How did I get the first one? I don't even know. What is going on? This is like hurting my brain. I can't go back either. How do I get this one? Oh! What the fuck? I don't know how I got it. Okay, that takes you here too. Lol CP1. Yeah, you only get mods in my It's actually true. I think all of the people that I've modded in my chat have been people who make troll levels. Oh my god. Alright, what's story? Wait, hold on. There is a story level inside of a story level. What? What's the story? What is this? Oh, it's a face. I'm pretty sure this is a head. This is its eye. The little mushroom is like a little nose. All right, we're going inside. Okay. We're going in. Yeah, we're going. So it's a reference to it's a reference to Kieran Carl's Con Crud, the level. Okay, I see. This is the butt. We went in the butt. But alas, the butt was not what we were looking for. No mod. Dude, I wonder if this is like an alternate account of somebody who was submitting all of these other levels and they got salty that I didn't mod them. Oh god. Alright, a fucking course! Never gonna give you up. Never gonna let. Oh my god. Oh, was that it? Okay. Mod Orion. Never gonna give you mod. Oh, I just saw the little goal over there. Lol, CP1. This is hilarious. This is a really good meta level. If you don't see the face, see the blue thing is the eye, and the green ground right here is the hair, and then this is the black coming out of the mouth. Good. Ban. No mod, only ban. Alright, that was funny. Alright, whatever. What the fuck ever. This is the jankiest setup of all time. Deal with it. Deal with it. For those who don't know, I work in a science lab and I do some sciencey stuff. So I'm going to tell you all about it. Okay, alright. Anyway, so whoa! Alright, woo! Alright. What do I do in the lab? So a lot of the work that I do in the lab revolves around this really cool type of bacteria. This guy's been around for a long time, so it's called Agrobacterium tumefaciens. I don't even know how to say it. I don't even know how to fucking say it. But basically, this thing is amazing. So this microbe lets you genetically engineer plants, and it kind of does it naturally too. Let me get a little cursor here. Woo, there we go. So like this bacteria causes these tumors on plants. So this is like a tree branch or whatever, and it's got these tumors growing on it. It's called crown gall tumors. And people figured out a couple of decades ago that this bacteria can basically genetically modify these plants, and that's how it causes these things. And so here's like a picture of one of the cells, like 100,000 magnification. So it swims around and infects these plants. But there was a great insight into this when people realized that the way that they do this, that this bacteria does this, can be modified so that you can put basically whatever genes that you want into this bacteria and it will give those genes to the plants. So like you don't have to cause a tumor in the plants, you can take out the tumor causing genes and put in whatever the fuck you want, literally whatever you want, and then you can transfer those genes to plants to make transgenic plants. And that's kind of how all the original transgenic plants were made originally. So the way that works, I'm not gonna get into like any scientific detail really, other than like, the bare minimum. So if you do want me to talk about anything specific, let me know. But basically, there's a part of this bacterium that like it has a bunch of genes and it attaches to the plants and injects those genes into it, and then those genes will actually like go into the chromosome of the plant, so it genetically modifies them. Here's a little electron microscope. Electron microscopes are super cool, by the way. Image of that happening. Does the plant give consent? Probably not. Yeah, so people figured out, hey, you can take out these genes that cause tumors. Oh, by the way, the reason it causes tumors is if you're just curious, the bacteria basically makes them produce like nutrient blobs. Like the tumors are food for the bacteria that it eats. Yeah, so here's a bunch of these little agrobacterium growing on a plate. You guys have maybe seen these things before. This is just like a nutrient agar plate, and the little dots on it are the different types of the little colonies of bacteria. They grow in these little circles that kind of expand, and they make these little colonies. And this is what it looks like growing in liquid. It literally, like almost every bacteria looks the same growing in liquid. Yeah. So this is where the fun begins with what we do in our lab. Oh, real quick here. An interesting thing also, just about like working in a lab and working with bacteria is like, turns out there are bacteria freaking everywhere. So a lot of like your job as a microbiologist is just growing the thing that you actually want to grow and not growing whatever the hell this guy is, though it looks freaking awesome. So yeah, that's a little bit of lab spaghetti right there. Yeah, so when people realized that this bacteria can genetically modify plants, that led to an entire field of called kind of like plant biotechnology. So check out this video. So this is us. This is one of my students. How do I? Okay. Can you guys hear this? So he's poking a tiny little dot in the plant, and then. Let's see, he's injecting a solution of these bacteria into the plants. Whee! No, that's not Joe. That was someone else who was on my PAX panel with me, though. Brandon. So yeah, you can just do something as simple as that to inject the bacteria into the plants, and then you can give them whatever gene you want. So like literally, it's up to your imagination to decide what gene you would want to give them. And I'll tell you about some of the stuff that we do to make vaccines doing that. But like commercially, you can literally just take this like giant ass thing of these plants, dunk it into a solution of those bacteria, and then all of these plants will gain those new genes. So that's kind of the point of that. Whoa, go ahead. All right, so like here's a plant leaf that I did that to that's producing a gene that makes it glow. So this one glows green, this one glows red. And a lot of the work that I did in the lab initially was mainly to try to figure out like, well, how do we convince the plants, genetically speaking, to produce the types of genes that we want, and also to produce a lot of them too, because if you're trying to make like a medical treatment or something, we want them to produce a lot of it. So we use these glowing proteins because or glowing genes because it makes it really easy to see what's going on. Demonic leaves. So we got some demon leaves. So anyway, here are the plants that we use. We grow them with these little seeds in these little containers. They're pretty cute. So this is when they're really tiny little babies, and then they grow bigger. This type of plant is a relative of tobacco. In the news, whenever the news covers this stuff, they always say tobacco. It's actually not tobacco. It's just a relative of it. It doesn't really have a good name though. Like it's Nicotiana benthamiana. We just call it benthy. But yeah, it doesn't really have a simple name. Here are my little plant babies. We grow them in this growth chamber. I think they're native to like Australia or something like that. Tobacco. Oh, here's me playing with some seeds. Whee! I made a transgenic plant, and those are me getting the seeds from my new transgenic plants. Whoa, go ahead. Okay. So one of the cool things that I get to use during our research is this thing is called a fluorescence microscope, and it allows you to kind of see those glowing plant leaves, but on a single cell level. So it actually lets you see the cells as they're glowing. So this is like 100 times magnification, and this is what one of the plant cells looks like. It's kind of crazy, right? It looks like a jigsaw puzzle piece. That's because all the plant cells in the leaf are kind of like, they kind of interlock like that it like gives them support and structure. It makes them stronger. So you can only see this one cell in this picture right now, But, well, actually there's two of them right next to each other. But actually the areas where like the jigsaw pieces would fit together, they're all full of cells. They're just not glowing green because they're not infected with these new genes that we've given them. The little dot is the nucleus of the plant cell, by the way. And then, you know, they just got these crazy shapes. Some of the work that I did also kind of we were studying ways of using plant viruses to sort of propagate the genes that we put into them. So you put in a gene, you put in this green gene with the virus, and then as the virus spreads between the different cells, you can see there's more of them infected here, you can sort of watch the virus spreading throughout the plant, giving it these new genes over and over again as it spreads. So this is kind of a cool way to do that. It's really cool looking at these. I love looking at these. They look really neat. Whoa, all right, shit's happening. So, all right, how to gene. Again, I'm not gonna get too technical with any of this, but like, I just want you, this seems highly unethical. What? They're plants. All right, so you might wonder, and this is something that I wondered for a long time, like how do you get all these different genes? Like how do you get the genes that you want to use? How do you transfer them around? How do you give them, this is already too technical, fuck, sorry. How do you do all of that? So we use these little molecular machines called enzymes. And this particular one is part of a process called polymerase chain reaction, or PCR. But this is just sort of what it looks like, you know, as a model when it's taking DNA strands and making more of it. So basically, this is a technique of making a lot of DNA. And that's all you really need to get out of this. PCR is like one of the most fundamental and amazing and powerful techniques of molecular biology that has been invented in like the last 50 years. So it's a super, super powerful technique because it can take any section of DNA and make a buttload of it. Like even if you just start with one copy of DNA, each time you run it through these little, this reaction to copy it, it doubles. So you get like 2, 4, 8, whatever. Skip a few, literally millions. How do enzymes know what to do? Billions of years of evolution. Yes, it's like an actual micro machine. Exactly. Is that my boy Taq polymerase? Well, this is just a general DNA polymerase. But yeah, the idea would be with Taq. I wasn't gonna get into that thing. That lagging strand ligation though. Yo, my bio majors, I see you out there. I see you. Yeah, so that's how police can get DNA from crime scenes instead of needing a bucket of blood. Yeah, and you can do a lot of crazy stuff. So you could like swab your cheek, and with just that little tiny cheek swab, you could get millions of copies of any human gene to then work with in the lab. You can also do I have some slides for this shit? Yeah, okay. So companies these days can make DNA very cheaply. So like these are so in order for, stepping real quick 1 second here, in order for this little machine to know where to copy from, you have to you can basically, without getting into any technical details at all, you can basically give it a little starting point so it knows where to start copying. And the whole point of that means that you can copy anything that you want. You can copy like just one gene, just another gene, just a part of a gene. And these little things are sort of like, a company can just send you these for like $6 now. It's so cheap. And you can use this to copy any DNA. So you could use it to, you know, for figuring out if someone's DNA is present at a crime scene. You can do a lot of other crazy stuff with it too. Random dumb Carl interlude. This is me staying amused in the lab. All right, anyway. All right, but yeah, in order to program in where you want this thing to start copying DNA, you need to know what the DNA sequence actually is. And another really fascinating thing that's been going on in the last 15 years is the cost of gene sequencing. So the original human genome cost $100 million or more, really, when they sequenced just one human genome. And the cost per genome since then has just been massively plummeting. This is a logarithmic scale, all right? We're from 100 million to $1,000. So this is Moore's Law, and it's faster than Moore's Law. Actually faster than Moore's Law by a lot. So what happened in 2008. It's called next generation sequencing. There's lots of new technologies that were developed to make it just so that you just like put in tiny little bits of DNA, and also like computing processing really helped too. Because really what happens is like you have billions of, we call them base pairs of DNA in your genome. And in order to like put them all, so when you sequence them, you just sequence like small little bits, and you need like it's really computationally heavy to kind of stitch them all back together in the right way. And so all the advances in computing power really led to that, making that a lot easier. So yeah, that on the other hand, that coupled with just how easy it is to do, makes this technique super powerful. So for example, let's say you find a sweet-ass jellyfish, and this sweet-ass jellyfish glows green under UV light. This is really cool. It's like Achoria victoria or something is the name of this jellyfish. Well, you can literally grind up some of that fucking jellyfish, toss it in a tube with these tiny molecular machines, sequence it so you know which is the gene that makes it green, and then get millions of copies of the glow green gene. And that's exactly what we did, and what people did to make those green leaves that I showed you back before there. Also, you can detect whether somebody's infected with something using this method too. So let's say you grab somebody's blood. This is like showing you this blob right here, these like bands of light, are the DNA that is copied by the polymerase chain reaction. And so like you can ask the question, is there a certain type of DNA present in this? So let's say you get somebody's blood and you want to know, does it have influenza genes in it? In other words, is that person infected with influenza? Jellyfish didn't ask for this. I'm sorry, jellyfish. So like you can tell, this isn't just a way of like copying genes and getting genes. You can also use it as a diagnostic to tell if somebody's infected with something, because viruses have genes too. And you can ask the question, is there genes from any virus in you? And so this is, you might recognize H5N1. These are genes from influenza virus. And so this sample tested positive for influenza virus because it matched these controls or whatever. So this is a way that people detect whether a virus is present somewhere. And it's so powerful because it needs so little DNA. Like if a fucking bat that was like infected with Ebola pooped like in an area a week ago, it might be possible to figure out that there's Ebola virus there using this technique. So again, just a really cool technique that lets you do all sorts of cool things, from gather new genes. You can even like put genes together with it in new ways, just detect stuff, all sorts of stuff like that. So what it actually looks like when you're doing some of this stuff, we have these like, I don't know how to describe it, but it's like this squishy gel. And when you put the DNA in it and put it through this machine that like puts it through an electric field, it allows you to separate out different pieces of DNA. So you can like, this is me with the little tweezers grabbing the piece that I want for this particular thing. And you can you can cut and paste stuff. These are like the cut and paste tools of genetics. You can be like, hey, I want to take this piece of this gene and that piece of that gene, and then you find your little machine that puts the two pieces together, that kind of stuff. So here's me in the lab being a fantastic scientist, adding these it's kind of weird because when you're doing this work, all of this stuff is like, it's so cool, but it's microscopic. Like you can't see any of this happening. Where's your lab coat? Fuck off. All right, anyway, I'm gonna delete that picture. Yeah, my lab is actually super safe. So, dude, exposed. Where are your safety goggles? Shit. Banned. I'm gonna get fired. Do you use lithium bromide? Yeah, or CyberSafe. So this is a graphical representation of a whole bunch of genes that we use, that we then give to the agrobacterium and then they give to the plants. So I'm not going to talk about basically anything on it besides that, just to let you know that like this is sort of what we work with, and we like cut pieces out of this and add other pieces and that kind of stuff. And then we give it to the agrobacterium and they put it in the plants for us. Now, just to note, there are a lot of other ways of transferring genes to stuff. The reason that the agrobacterium is really nice is just that bacteria are cheap as heck to grow. So it's just very convenient and cheap. But there's definitely other ways you can do it too. All right, another cool thing about this is like when we make these agrobacterium, you can do this with lots of different type of bacteria. So this is a freezer that gets down to minus 80 degrees Celsius, which is like minus 112 Fahrenheit. So it's really freaking cold. And if you put the bacteria in this, they can actually survive for basically centuries. They can survive a long ass time. And so once you like make one of these new combinations of genes and put them together into one of these bacteria, you can just store them in the freezer forever. Like we have stuff in our lab that I'll pull out a tube from like before I was born, and I'm like, Jesus, still alive. So yeah, the half-life of DNA is like 500 years, so no Jurassic Park, but stuff still. How do you stop cells from bursting? So there are cryoprotectants that you can put in. We use glycerol. Basically, I didn't prepare a slide for this, but like when you put a bottle of water in the freezer, you know how it like expands outward? The same thing happens to cells, which is why you can't just freeze people. I mean, there's other reasons too, but like you can't just freeze shit because the cells will literally like, the liquid in them will bleh and blow them up. But you can put different chemicals in to kind of stop that expansion from happening. And that works great when you're a single cell. It doesn't work so great when you're a human. So it's easy to protect bacteria from freezing, not so easy to protect humans. Anyway, it's pretty cool. Oh yeah, speaking of cold stuff, this is pretty fun. This is very, very high quality science going on right here. This is an inflated glove in liquid nitrogen. The gas itself contracts when it gets really cold. And then when it warms up, it expands again. Whee! All the lines in it went away. Pretty cool. Liquid nitrogen is really fun. Alright, anyway. Carl easily amused. Okay, yeah, so, right, so we can get genes, we can cut and paste them, we can put them in this cool bacteria, we can transfer those genes to plants. But a lot of the work that I did studies genetics at sort of a more basic level, because it's like, I've always found a popular misconception, when people talk about genes, it like infuriates me when people are like, oh, you know, the gene for X, the gene for happiness, the gene for obesity, and like, all that shit is like so ridiculously oversimplified and dumb and impossible. Like, that stuff really, really annoys me. So genes are so much more than just one thing. So remember I told you there's that jellyfish gene that glows green. So here's the green glowy jellyfish gene, and here's the same gene, and here's the same gene, and here's the same gene, all put in different spots on this plant leaf. And you notice how they're very different amounts of it that are being made. So one of the things that I studied was how organisms control like how much of a gene to make, when to make it. Do you only make it like when you're sick? Do you only make it when you're sleeping? Do you only make it, you know, a little bit in the morning, a little bit more at night, all that kind of stuff. So genes are like a lot more complicated than just like a gene that produces an effect. And I could go on a whole giant thing about that, but yeah, I'll skip it for now. But so a lot of the research that I did was just kind of studying that. And for the purposes of producing useful things like vaccines, we want to kind of understand how to convince the plants to produce a lot of the thing that we want. So this guy was more useful to us than that one. All right. Oh yeah, so another thing is like genes function differently in different organisms, though it's amazing how similar they do function in different organisms. So like this is lettuce, this is spinach, this is tomato, and some of the experiments that I did were sort of studying how these same genes and these parts of the DNA that control how much and when and whatever of these genes is made, how those function in different species of plants. So I grew up all these different types of plants, tested them in those too. Eat your lettuce, Carl. Okay, anyway, so let's get to the vaccine stuff. So once we kind of have this system of making genes in plants, one useful thing to do with it is to make vaccines, because plants are really safe, they're really cheap to grow, and so they're really useful because like if you want to make a vaccine, you kind of need to like get a whole bunch of maybe a part of a virus or something like that. And it turns out it's not very ethical to just like farm humans for tons to like infect humans with viruses and then farm them for them. Not gonna fly. So plants work a lot better for that. Okay, so I'm just gonna cover very, very briefly like what is a vaccine and what is a virus, because if you don't understand that, none of this is gonna make any sense. But basically viruses have a protective coating around their genes on the inside. So like this is the genes on the inside, this is the protective coating, you know, in graphical form. And the protective coating is sort of how they get to where they need to go, but it's the genes really that make you sick on the inside. So when a virus will like give its genes to you and sort of reprogram your cells to make more viruses. So one way, the history, I'm not gonna get into like, I don't wanna get into like the whole history of vaccines, but lately we've been trying to make new types of vaccines that don't use living viruses for various reasons. And there are some viruses that lend themselves really well to doing this, because some viruses, they pretty much will make only one gene that sort of like sticks to itself many ways, many times to make that protective shell. So basically you have this like complicated three-dimensional protective shell thing. It actually makes an icosahedron, by the way, so hey, where my board game nerds at? That's what the virus might actually look similar to. And all of that is just one gene that comes together in all these different ways to make this. So why do you want to do this? Why do you want to make this shell of the virus? Well, this is more like what it really would look like, or you know, a model of it. And so if you look at this one, this is our fake empty virus. It's just the protective shell. There's no genes on the inside. In other words, it can't make you sick. And this is the real virus. It has the genes on the inside and it can make you sick. Can you tell any difference between the two of them? No, neither can your immune system. And that's the point. You can make something that looks like a virus, but it can't make you sick. Whereas the real virus with the genes on the inside would make you sick, but your immune system can't tell the difference. Slightly smaller, fuck off. They have different words underneath them. I was having a midlife crisis. So these things are called virus-like particles, is what we do. So we make these virus-like particles that resemble real viruses, and then you give them to somebody as a vaccine. This is already how the current HPV vaccine works. Yeah, I think it was the first vaccine that was approved using this kind of method. Most of the older vaccines use actual live viruses that have just been weakened. All right, so how do we actually do this? Well, we get the genes for the virus using the methods that I talked about. Like you get the little molecular machines to amplify a whole bunch of them, cut them up and paste them into the agrobacterium. And then these agrobacterium you put into the leaves, and you know, you can't see it this time, but interestingly, the leaves like get kind of shiny like this, which is, I don't know why it happens, but it's cool. So get a whole bunch of these leaves, and then you blend them all up into a vaccine smoothie, as I like to call it. And then you do a bunch of chemistry on it. So here's a little solution of that. So this thing is like ground up leaf smoothie, but it's full of those virus particles that I mentioned, that like the vaccine thing. So this is just doing some chemistry on it to remove parts of it that we don't want. We put it in this device called a centrifuge that spins it really fast. And you can see like this part right here is like all the green stuff in the plant. And then this part that has the virus particles in it, we use because it's a lot cleaner now. Centrifuges are pretty neat. And then we do more biochemistry that I'm not going to get into, but basically like we find, there's a lot of different techniques that you can do here, but the essential bottom line of it is it kind of filters out a lot of the plant proteins that we don't want and just keeps the virus proteins that we do want. So here's a little dripping thing of that going on. Essentially this thing filters out the stuff that we want. Then we put it in this little apparatus here. It's similar to what we do with the DNA, but this one is like, it takes all of the proteins and it separates them out, and it's sort of like a method of analyzing proteins. So you might get something that looks like this. This is like an actual gel, and the little like blue bands in here are all of the different proteins that were in the plant cell. And so you can use that to analyze whether the protein that you want is actually being made or not, that kind of stuff. So if you just look at this one right here, I'll very simply explain what the hell this is. So this is a representation, an actual, like these are all the actual proteins that the plants were making. The one in the middle here, see this big like blob? That's rubisco. It's actually the most abundant protein on the planet, and it's what plants use for photosynthesis. But when we give them these new genes using these methods, so like remember when I made the green one? That's this one right here. So you can see that like these plant leaves, about like almost half of all of the protein that they're making is the new gene that we put in. Or in this case, this one right here is making norovirus gene. So this is the empty shells of the norovirus. This is this big fat blob right here. And then this one is making an antibody called rituximab. It's a drug that is used to treat B-cell cancer. So this antibody is used for that cancer treatment. And all of these were made in these plant cells using all of the methods that I talked about. And again, it's literally up to your imagination to try to figure out what you would want to make with these plants. That's one of the cool things I like about this, is there's so many possibilities. It's like, you know, we can make cancer treatments, we can make vaccines, you could probably make a lot of other stuff too. All right. Anyway, one of the other things that we do to try to figure out if all this stuff is working right involves like, it's kind of cool. It's called a Western blot. But basically you end up in a dark room like with this film, and on this film will make like only the particular gene that you're interested visible. And it's really crazy. Like when I started to think about it one day, I was just like, I'm just sitting here in a dark room, like looking at these smudges on this thing, trying to like read the secrets of nature. It sounds like complete mad science. It's really cool though. It's really crazy and really cool. So it looks like this, and it's like, but one of the weird things about being a scientist is it's like, if one of these little bands of darkness is like in the wrong spot, you might have a bad day. Like your day might literally go to shit based on whether one of these things is in the wrong spot. Also, lab spaghetti happens too. This thing over here is like, when if massive lab spaghetti happens, you might end up with this. This is a bad looking Western blot. And over here we have a nice looking Western blot. So it basically tells us whether the genes that we wanted to put into the plants are all being produced properly, are the proteins that we're trying to make being formed correctly. It allows you to answer questions like that. So another fun device that we get to use is an ultracentrifuge. So this is an even bigger, crazier centrifuge. So this is like some plant extract over here, just like ground up plant stuff. And we spin it in this thing at crazy, crazy high speeds. This thing can do like 250,000 times the force of gravity. So it spins very, very, very fast. And you can see all of these, like all the stuff that was at the top will form these different layers down here based on how dense it is. So this allows us to separate different components of the plant cell and pick out just the ones that we want. Turns out all the viruses go to one spot, so that's really useful for us. It allows us to gather out all the viruses. And then this is probably the coolest thing that I got to use in grad school. So this is an electron microscope, and it allows you to actually image extremely small things. So like, I don't know if you guys know how small viruses are, but viruses are really fucking small. So with a normal light microscope, you can't even see viruses. You can see bacteria, but not viruses. And so these are some of the little viruses that we made. This is at like 400,000 times magnification. And if you remember this thing, this is basically, let me do this. Whoa, I killed it. I killed it. This fucking thing right here is basically one of these. So you're trying to envision it. Sort of one of those things. Okay. So then once we study, purify, and like, you know, make sure that our vaccine is being made the way that we want, and we've gotten a lot of it, then we actually test it to see whether it works as a vaccine. So here's me delivering the vaccine to be tested, you know, as Carls do. Oh god. Yeah. So it gets injected into mice. And we test whether or not the vaccine actually works. What the? All right. So after vaccinating the mice, just a fun fact about blood. Sorry to throw some blood at you guys. But like, so blood has two components, and in the if you like spin this thing also in a centrifuge, a lot of the clotted blood goes to the bottom, but then you get this like clearer stuff on the top for blood serum, which when you don't, if you ever donate blood, sometimes you may just donate this, and then they put that stuff back. Just, I don't know, fun little blood facts. We essentially are looking for whether or not the mice have produced, whether the immune systems in the mice have made, does people know what antibodies are? Like how, how common is knowledge of what an antibody is? I don't actually know. It's like sometimes you just forget what is common knowledge and not. Okay, yeah, sure, everyone knows what an antibody is. Okay, yeah. So basically, we're trying to make antibodies, which are just like parts of the immune system that help protect you against stuff. I don't know. There's my 2-second description. So this is a test called an ELISA, and it essentially will tell us how many antibodies the mice are making against the particular virus that we're trying to protect them against. So just you use these different equipment. We can test like all these different samples at once using this little plate. I'm not gonna bore you guys with the details. So these are called pipette tips, and pipettes are like little devices you use to transfer around liquid. And this is when you have a bad day. This is lab spaghetti right here. This is lab spaghetti because there's so many of them and they all fall out. Rip. This is me looking at the results of one of the ELISAs. We got these cool little like pipettes that'll do, you can do like 8 of these different samples at once with it. It's kind of fun. And here's what the results might look like. So on this side of the plate, these represent 6 different mice. So like these 2 things are from one mice, this is from another mouse, this is from another mouse. There's 6 mice on this half of the plate and 6 mice on this half of the plate. This one on the left was like the old version of the vaccine. Oh yeah, and the bluer it is, the more antibodies the mice make. So basically, the bluer it is, the better for the vaccine. And then this was the new one that we tried to make to try to improve it. So this was a good day in the lab. This is a good day in the lab. Rip old mice. Yeah, do mice give consent to be infected? So we don't actually infect the mice with anything. We just, like, it's sort of the same way that people do these tests with humans first. You can still give somebody a vaccine and kind of like measure their immune system to see if it might be working without actually infecting them with anything. Yes, all of the mice got autism. No, I don't inject the mice. There's a vet that does it. Yeah. So mice go, hey Ari, male and female mice. Is that the actual science? In case you're wondering, by the way, when I first did this, like I'm a huge animal rights kind of guy. Like, I actually did not like, I don't like the idea of using animals for research if you can get around it. But so just if anybody's curious about that kind of stuff, like, cause I know I was when I was first doing this, all of these mice are like treated better than the students are. Like, these mice are extremely well cared for, and a trained vet does all of the stuff with them, and she fucking loves them. So yeah, in case anybody was worried about that kind of stuff, the mice are very well treated. Anyway, yeah, so I actually literally, this is not planned at all, literally today, my first vaccine paper on trying to make a new HPV vaccine was just accepted to a cool journal called Vaccine, which, as you imagine, is a scientific journal about vaccines. So literally today, they got the word this got accepted. It's not published yet, but when it's published, I'll post it on my Twitter if you guys want to read it and get into more of that science stuff. So anyway, I'd be happy to take any questions that you guys have. I didn't want this to go on too long, so I didn't prepare anything else besides that.